Hello, and welcome to this Nature Custom Media webcast titled How to Break a Fundamental Limit in Fluorescence Microscopy. My name is Alison Halliday and I will be your moderator. Today's webcast is sponsored by Hamamatsu. Fluorescence microscopy has revolutionised how we see and understand the working of living cells. But there is a major limitation. The illumination required to excite fluorophores leads to their gradual photodegradation. This phenomenon, known as photobleaching, limits both the duration and time resolution of experiments and degrades image quality. However, by adding an infrared laser to a fluorescence microscope, a simple modification costing less than $10,000, it is possible to more than double the number of photons that green fluorescent protein, or GFP, emits before photobleaching. The catch is that this infrared co-illumination trick works only in light sheet microscopy and not in widefield or confocal setups. In this webcast, we'll explore the best live cell fluorescence microscopy practices for achieving at least twice as much information per unit damage to the biological sample, helping to push the limits of fluorescence microscopy. You can access closed captioning for this event by selecting English, British at the bottom right corner of the video player. With us in the studio today is Faderao, Senior Engineer at Biohub. We will then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your question here and then press send, and we'll do our best to answer them at the end of the session today. And now, over to our speaker.
Hi, my name's Tanner. I work at the Biohub. I am a light microscopy engineer, inventor, scientist. I'm going to talk to you a little bit today about some recent innovations in live cell fluorescence microscopy for cell biology, and some innovations that I've made on top of that. But to give you a little background about things that I'm interested in, my PhD advisor, Paul Maddox, would always say, if you take an image of the ocean every second, you see the waves. But if you take an image of the ocean every minute or so, you see the tides. And I actually sat down and I went out to Point Bonita, north of San Francisco in California, and actually did this. So what you're seeing on the left here is a time averaged version of the movie on the right. So effectively, I'm imaging once every 3.5 seconds on the left, and you see this mist. You see this temporal averaging of air and water. And that to us looks silly because, of course, we know that that's actually waves and air. But if you've never seen waves before, like you're seeing on the right, imaged at 30 frames per second, then you wouldn't even know that it's there. And you'll notice that the spatial resolution of the two images is actually quite different. Not because I blurred the image at all, but because I am averaging something that happened over a much longer course of time than the actual event occurred at. And you might think, okay, so what does this have to do with cell biology? And I'll actually point out that low temporal resolution, when you're recording a live cell, actually will degrade your spatial resolution. And here's a real life example of that. You might be familiar with the endoplasmic reticulum in adherent tissue culture cells. Looks something like this. This is really well studied organelle, and it has, at least up until a few years ago, been described as having two distinct morphologies at the cell periphery. These tubules, which you can sort of see here, are these little spaghetti spindly like parts of the organelle, and then these sheets, these flat pancake-like structures that connect the tubules. This is an image taken at, we'll say, 1 frame per second at 1 hertz. But if you actually build a better microscope that is capable of imaging much faster, you end up noticing that there are these little gaps, these little holes in there. And when you image, at something like 40 hertz, you see that these holes are dynamic. They move around. They're moving around so fast that temporally they average out into something that looks like a sheet. And colleagues of mine confirmed via cryo-electron microscopy with correlative fluorescence microscopy that these tubules are real. And the sheet is simply an artifact of imaging too slowly. There's a lot of biology that we miss by not imaging quickly enough. And so that's really the focus of a lot of the work that I do. A lot of my interest is how do we build better technology to view an aspect of biology that hasn't been possible to view before, which is the time domain. There's a fundamental problem with imaging fluorescent samples over time. And I'm sure any cell biologist here that has done any live cell fluorescence microscopy knows this, that even after a short amount of time, your fluorescent signal degrades. This is photobleaching. This is what happens when you leave a flag out in the sun too long. The colors fade. Your cells, it can happen much quicker because the intensities of light that we're putting on these cells are much higher than, we'll say, sunlight. This in and of itself, while bad, is not completely detrimental because all we're doing here is making it impossible to look at something forever. We have a finite, we'll say, photon budget, the number of photons we can get out of a fluorescent sample, which limits the total amount of time that we can look at something at. But in live cell fluorescence microscopy, I'll point to a second and frankly much worse problem, which is that of phototoxicity. When you put a fluorescent marker inside of these cells, like for example GFP, and it photobleaches, the act of photobleaching can and often does release reactive oxygen species into your cell, and these are incredibly poisonous. Your cell can deal with a little bit of it, and depending on the cell type, some can deal with a lot, some can deal with a little bit. Regardless, what I'll often point to is that a little bit of photobleaching often results in a lot of phototoxicity. And so you often will begin observing a live cell and then immediately transition to observing a dead or dying cell. And it often reminds me of these old nature documentaries where you'd have David Attenborough style, narrator as they zoomed in on a caterpillar and said, "The humble caterpillar crawls along the leaf, consuming 3 times its weight, and soon it will form a chrysalis and emerge as a beautiful butterfly." And then they do a hard cut away from that because 10 seconds after they stopped filming, the caterpillar shriveled up, rolled over, and died. Because they had to put so much light onto that caterpillar. Film was so insensitive that they had to put that much light on there that they literally were killing it. So they weren't watching a caterpillar living, they were watching a caterpillar dying. And that's the main thing that I want to try to avoid when we're doing live cell fluorescence microscopy, is because to me, the time domain is so interesting that you can get out, because we don't observe the cell directly with fluorescence microscopy. We observe light coming off of it. And that means that we're able to look at it in multiple points in time. Assays like genomics or mass spectrometry, for example, physically, break the cell apart and measure the components directly, but you only get to do that once. Whereas with light microscopy, we have the ability to image through time. But we don't want to do that if we're killing the thing that we're trying to understand. So why can't we do this? Why is this such a challenge? And what can we do to sort of ameliorate it? And I'll go over some basic fluorescence microscopy. The most common, I would say the easiest, I would say, of fluorescence microscopy is the epifluorescence modality. I'll represent sort of the sample here as this gray blob filled with little tiny fluorescent proteins, GFPs. Typically, most cell biologists will mount their sample on a thin piece of glass, and then they will observe it here represented from this eye, basically underneath the cell through this clear piece of glass. And in order to visualize a fluorescent sample, you have to deliver excitation light, in this case blue, to the sample, and then collect the green fluorescent emission light from the cell. This, while mechanically and optically simple, is, in my opinion, one of the worst ways to do fluorescence microscopy for 2 reasons. The first is you only have a very thin focal plane of your cell in focus. When you use high resolution lenses, your focal plane is actually quite small. You're typically a fraction of a cell thickness, less than a micron. Your cell can and often is be a lot thicker than that one individual focal plane. And that means that even though I am illuminating pieces of the cell that are in focus, and that's the part of the image that's going to appear sharp and in focus on my detector, there are fluorophores above and below your focal plane that are also being illuminated and therefore glowing. So I could focus on my hand, or I could focus on the computer, and even if I'm focused on my hand and everything is nice and sharp and in focus, the light coming off of my computer screen still reaches my eye, but it's blurry and it's out of focus. So it actually will drown out in focus information of my hand that I'm trying to observe. Not only do you have the problem of out of focus emission, you also have the problem of out of focus excitation. I told you previously that every time that a GFP gets excited by a blue photon, there is a non-zero chance that it irreversibly becomes bleached, therefore decreasing your signal and poisoning your organism. So even though I'm trying to observe only these two little particles in the focal plane of my sample, I am damaging every part of my sample trying to do this. So to improve upon this, the confocal microscope was invented, and this is by no means a recent invention. I believe Minsky first published his confocal microscope in like 1957 or so. But you go into any fluorescence microscopy core, and the confocal microscope, be it a laser scanning confocal or a spinning disc confocal, is typically like the staple. You'll see one, if not multiple instances of this, and for a good reason. It is a pretty strict improvement over the epi fluorescence microscope, or often called the widefield microscope. And it solves one of the two problems that I mentioned previously. It solves the problem of out of focus emission, because through the clever use of focusing the light and refocusing any emission light through a pinhole that is at a conjugate focal plane, it rejects light that comes from fluorophores above or below the focal plane, which is really great if you're trying to do 3D fluorescence microscopy, because now you're seeing not a projected view of some in-focus fluorophores and a bunch of out-of-focus fluorophores. You're only receiving light of the in-focus fluorophores. However, this doesn't solve the problem of out-of-focus excitation. In order to excite fluorophores in the focal plane, I have to pass this light through fluorophores below the focal plane, and therefore it also leaves the cell by exciting fluorophores outside of the focal plane. So I'm still generating emission light and therefore photobleaching events outside of the focal plane, but I'm rejecting the light that comes from them. So this by no means decreases the amount of photobleaching or phototoxicity that you're doing to the cell. So another improvement that got made is this thing called light sheet fluorescence microscopy. And light sheet by itself is no recent innovation. I believe the original paper was published by Zygmuide in 1904, but its application in fluorescence microscopy was only realized in, I think, 2004 by Jan Huysken and Ernst Stelzer. So it is fundamentally different from confocal because it only excites fluorophores at the focal plane of whatever lens is observing the cell. So it's not that we're exciting all of these fluorophores and rejecting the light that doesn't come from the ones that we don't want. We're simply not generating it in the first place. And this massively increases signal to noise as well as decreasing photobleaching and phototoxicity. So it really lets us damage our cell a lot less. And so every time that we take an image, it costs us a lot less signal. It costs us a lot less cell viability, and it therefore allows us to image faster and longer. To show you some real world examples of this, on the left here, you're seeing a human endothelial cell expressing a construct called GFP-EB3, which shows the plus end of polymerizing microtubules. And we're showing it to you in 3D. In this case, I'm showing you two orthogonal projections, the XY maximum intensity projection and the ZY projection. And what I hope you can appreciate is that In a spinning disk confocal, the cell almost immediately bleaches after only a handful of volumes. I'm showing you a very similar cell taken on a light sheet microscope on the right. And you're noticing that even after 100 volumes, it has only bleached to a fraction of what the spinning disk microscope has. And in fact, the spinning disk movie cuts off because they didn't want to continue imaging it. It was basically completely black, and I believe the cell died. So, hopefully what I'm impressing upon you is that light sheet microscopy is not only gentler, it's also faster. And this is a particular point that I think is not often talked about, or not talked about as often. With confocal microscopy, you are imaging one point or a series of points at a time, but those points by definition have to be sparse. So you're not imaging an entire plane at once. You are imaging, just a subset of that plane. Whereas light sheet, because we can expose the entire plane at once, we have a much higher duty cycle, much higher, basically, like efficiency of collection, and we're able to image a lot faster. How much faster depends on basically how slow your confocal is. But if you think of an image as being just a grid of 1,000 by 1,000 pixels, a laser scanning confocal takes 1 pixel at a time, whereas a light sheet microscope using a camera takes all 1 million pixels at exactly the same time. So if light sheet is this much better, then why is it not the staple in every core for live cell fluorescence microscopy? And the answer is it's really annoying to use, at least in the traditional sense. So light sheet microscopes, as they are currently built, or not currently, but as they have been built since the last 20 years or so, require this frankly odd 2-lens setup where you have an excitation lens that is at a right angle to your detection lens. And this makes sense. You want to deliver a sheet of light at a 90-degree angle to your detection lens, because your image plane for your detection lens is here, and you want to send out a light sheet with that. And while this is optically more simple because the detection path is effectively just a very basic microscope, this makes it a lot harder for biologists to image the samples that they're used to. I showed you before these fluorescent samples as being mounted on cover slips. That is when I was doing cell biology, that is how I mounted all of my cells. That's how 95% of all of the biologists that I interact with prefer to mount their cells. And this 2-lens geometry is not particularly amenable to that. You end up needing to create a media bath where these lenses physically dip down into your sample media, because you need a continuous optical path, not for one lens, but for 2 lenses. So forget about using any sort of high throughput imaging with this on a plate with multiple wells. You have to switch your sample out each time and fill it with typically several milliliters of sample. So not only is it a little bit annoying, or extremely annoying on the biologist side, I'll also point to some optical constraints that this geometry imposes. I'll talk a lot about numerical aperture today, which is effectively the light gathering efficiency of the microscope, and how the geometry of these lenses, or the geometry of light sheet microscopy as it was traditionally done, prohibits the use of what I'll call high NA lenses. And just a brief sort of introduction to numerical aperture. If I have 2 lenses, one of which is a low numerical aperture lens on the left, one of which is a high NA lens on the right, and I have 2 point sources that are basically emitting light in all 360 degrees, 4 pi steradians. Some of that light is going to be captured by the objective lens. It should come as no surprise that the more light you collect, the better it is, not only because it forms a brighter image, but also because it forms a higher resolution image. And this is because the more angles of light that you can view a sample from, the more precisely you can localize where that light was emitted from. So these are 2 really fundamental parts of why high NA is desired by microscopists and cell biologists. But like I said, you can't have a high NA lens because you would need two of them, or you would need at least one high NA lens and one low NA lens that would need to physically not like touch each other. So the presence of this second lens really restricts the effective resolution and light gathering efficiency of your detection lens. So while imaging through the cover slip is easier on a biologist, they can just bring their sample over, slap it down, because your sample is now distinct, like from the microscope itself. It's more damaging than light sheet. So you have this like really unfortunate trade-off where your lenses are dipped into your sample fluid in light sheet. You restrict your sample dimension because scanning is a lot trickier when your microscope is literally dipping into it. Your numerical aperture is lower in light sheet, but it has the one extremely nice benefit is that it's much faster and much more gentle. So you can image for much longer or much faster. So the bulk of my scientific career has been, how do we combine these two? How do we get the advantage of light sheet that is fast and gentle, and the ease of use of a single objective lens mounted next to a normal sample that a biologist could find easy to use and potentially in a more high throughput way. So here's the problem with that, though. If I have a single lens, I can make a light sheet with a single lens. That light sheet can be tilted to a certain extent. I can't tilt my light sheet so that it is perfectly flat. This is why you need, in traditional light sheet, a second lens over here. These lenses have a finite angular range that they can deliver the light at, and the focal plane of that lens is at an angle that is basically forbidden. So if we can't take a light sheet and make it agree with our focal plane, then let's tilt the focal plane. Let's change the plane that's imaged onto our camera to match the light sheet. Easier said than done. This has been the focus of several very smart people in the community for several years now. I'll take you through a brief history of that and bring you to where we're at today. So oblique plane microscopy, published by Dunsby in 2008, was, I think, one of the first examples of what I'll sort of broadly call the single objective light sheet class of fluorescence microscopes. So he also realized this problem of, well, if your focal plane is parallel to your lens here, and you can only excite your sample with a tilted light sheet, how do we untilt that image plane? And he figured out a clever way of doing that with the use of two more objective lenses, and here L6 and L7. I'm going to, for the rest of the talk, sort of refer to these as objective 1 or O1, objective 2, O2, and objective 3, O3. You'll notice that objective 3 is tilted with respect to objective 2. And that's by design. That tilt angle actually matches the tilt angle of the light sheet, because what we do is, with this second objective lens, we reform a remote image of our sample in air, a far distance away from our sample, and then we re-image that view at a tilt, so that all of the photons that appear on our camera via this third microscope here, objective lens, tube lens, camera, all of the photons that appear on that chip are in focus, and we don't have to reject any that are out of focus because there are none. So great. Why aren't we all using this? Here's one of the trade-offs. If I tilt two objective lenses, and these are high numerical aperture objective lenses, if I tilt them relative to each other, and you look at, okay, here's the light that comes from my sample that is delivered by the second objective lens, and then what is the light that is collected by the third objective lens? It collects a portion of it, but there is a large portion of it that is lost due to the fact that this third objective lens's cone no longer fully overlaps with the second objective lens's cone. So not only does this lost light result in lower detection efficiency, a lot more photons are lost because they're basically just sent out over here. It also results in lower resolution. You're viewing your sample from fewer angles. You are less certain of where those photons arrived from. You have lower spatial resolution. And while for some applications that might be acceptable, I care deeply about, every photon is precious. Any information spatially that we can, there's money left on the table. And so how do we take advantage of that? So Bo Wong and Bin Yang came along in 2019, and they presented the Epi Selective Plane Illumination Microscope, or eSPIM. SPIM is another acronym that you'll often see for light sheet fluorescence microscopy, a selective plane illumination microscopy. This was particularly clever because what they do is exactly the same thing that Dunsby did, where they deliver a light sheet at an angle relative to the cover slip and the focal plane, the native focal plane of objective 1. But instead of using another air objective lens for objective 3, they actually use a water objective lens. And there's a really cool reason for this. You might know that, an objective lens that has an immersion material that it expects that is a higher refractive index than air, things like water immersion objective lenses or oil immersion objective lenses, can have a higher numerical aperture than an air objective lens. And that's because of how light refracts, how light bends when it goes between different materials of different refractive indices. When light goes from air to glass or air to water, it bends. And what they did with this water objective lens is so clever. They filled this gap between the objective lens and the cover slip that this objective lens requires with water. So basically, this lens doesn't work unless it has exactly the right amount of water and a cover slip in front of it. But that lens has a working distance, and the working distance they set as they set the length between the cover slip and the front surface of this objective lens as the working distance of the lens. So its focal plane was at the surface of this glass cover slip. And that's clever because this lens is perfectly capable of collecting a unaberrated image at that particular focal plane. It won't do it well if it were further into the air, but it's not. It's right at that surface. And this light that was delivered by objective 2 now bends when it enters this higher refractive index, and it actually compresses that cone such that basically all of the light that is delivered by objective 2, with the exception of a small sliver here, gets refracted into objective lens 3 and detected. So now what we've got is a single objective light sheet microscope that doesn't waste photons, which drastically improves your resolution and your light gathering efficiency. And then my buddies Andrew York and Alfred Millett-Sicking came along later in 2019, sort of inspired by the eSPIM, and created a solid state objective 3. This is, I'll argue, a strict improvement over the water immersion lens, because you could buy that off the shelf, but you have to keep a cover slip exactly the right distance away from it and keep it hydrated, fill it with water. Whereas this is, at the time, a custom objective. Now you can buy it off of the shelf. And what you can think of like this lens, and you'll often hear this referred to as the snout lens or Mr. Snouty, or the microscopes that are built around it as the snout scope. That's sort of what it's called in the community, because you'll notice that up until this front surface, it looks like a normal lens. And it is. It's a normal oil immersion lens. It's just that they have glued on it a chunk of glass that is exactly the thickness of the working distance of that objective lens. And then they've ground off one side of it so that this lens can be tilted. And so what you're left with is an objective lens that has a 0 working distance and a numerical aperture of 1.0, which is actually a modest numerical aperture for an oil immersion lens. You'll often see oil lenses having NAs of up to like 1.45 or even 1.49. So NA 1.0 is actually pretty modest. But if you look at what light it can accept from this second objective lens, it can accept light that is traveling up to 90 degrees relative to its surface before it refracts in. And what this lets you do is build basically an entirely solid state, stable, what we'll call single objective light sheet or SOLs. And I'm showing you a diagram of like how exactly the microscope sort of functions. But effectively, you bring in a light sheet that you scan laterally through your sample. That light is collected by your objective lens, delivered, descanned by this same galvo mirror, just like it does in laser scanning confocal, refocused onto this remote image plane, where it is viewed from a tilted angle by the solid state objective 3, the snout lens, before being refocused onto the camera. So the image that you see on the camera is a tilted slice of the sample at any one point in time. I'll plug the Snout Club that Alfred is maintaining. There are 80 plus instances of these microscopes around the world. You can follow this QR code to see the latest updated list. I've built 4 of them at this point. They are it is the best way to do live cell fluorescence microscopy, and I'll pitch some data to support that. But if you're at all interested in trying out one for yourself, search this list. I bet there's one somewhere near you. We're trying to put them more in cores that are more accessible to, like, the average cell biologist. And I'll talk about my efforts doing that. So this is a sped up time lapse of me building one of these single objective light sheets at UCSF at the Center for Advanced Light Microscopy. This was taken, I think, over effectively like several weeks. It's really just me moving my mess back and forth around my optical table. I don't expect anybody to like follow assembly instructions based on this. But they're not that difficult to build. The only tricky part is getting the 2 objective lenses, like O2 and O3 aligned. And we're able to teach people how to do that in about a day. Side note, I really enjoy teaching technically minded people or biologists to build their own instruments. So if that's at all interesting to you, please reach out to me. I'd love to have an alignment session where I teach people the right way to do optical alignment, and especially the 2 objective lenses. So to actually go into some of the data that you get off of this microscope, what I'm showing you here are the raw slices of a tissue culture cell as the light sheet basically is driven through the cell. If let's go there. Yeah, driven through the cell. And you can see, if I pause it right in the middle here, the cover slip is this black box below. The cells in this case look like a fried egg. We're seeing the nucleus here. What I'm showing you is labeled as vimentin, particular cytoskeletal filament. And as this light sheet is driven through the cell, you'll notice that you get different slices of it. The data here, I'll say, look skewed, and that's by design. That's because the direction that we're sweeping the light sheet is not orthogonal to the Y dimension of this image. So it looks like the cell sort of like moves up. This is a relatively simple computational problem to solve. Basically, it involves taking each slice and displacing it by a certain number of pixels along the Y dimension. And when you do that, it's called deskewing. You get a, we'll say, normal looking image of your cell. If anybody studies vimentin, hope somebody can vouch whether the cell looks normal or not. This is just to give you a sense of like, what do the data raw coming off of the microscope look like? And how do you transform them into an image that a biologist might recognize as coming off of something like a confocal microscope? Now, I'll note here that this is a basically max intensity projection along the Z dimension. We certainly can show you the full 3D like stack. I'll show you that in hopefully a couple of slides. But representing 3D data on a 2D screen is hard. So this is what basically the de-skewing looks like, taking each of these slices, now in this case of a clathrin expressing, or a clathrin GFP expressing cell. And we can do this sweep several times through time. And again, showing you a projection. Here is what the time lapse of these clathrin coated vesicles look like. This slicing, as I'm sort of showing you, is way slowed down. We can take several hundred, if not several thousand slices per second, because that's how fast modern cameras are nowadays. And that allows us to, image an entire volume in basically less than a second. So what is this good for? Anything that is particularly light sensitive, because now for every two-dimensional image that you take, you only damage a two-dimensional slice of your sample rather than the entire 3D volume. So with these killer T cells that I'm showing you here in red, attacking these cancer cells in cyan, these T cells are particularly photosensitive. They do not do their search and find behavior where they're crawling towards the target cells or forming these synapses with them. They don't do that when they're being poisoned. They are very dynamic. They move quite a bit. We have to image them fast, and we have to image them gently in order to see this in 3D. This is the sort of data that you can get off of it. So it's not only gentle, it's fast. What I'm showing you here is a somewhat 3D projection of a cardiomyocyte expressing a calcium sensor. And this is being imaged at 10 volumes per second. So you can actually see the calcium wave propagating across the cell from along the processes from the nucleus outward. This is, we can take very short exposures because we're incredibly efficient with the light that we collect. We're not damaging the cells. So anything that requires like moving cells, things with muscles, things that swim across your field of view faster than you can capture, things that are particularly sensitive. That's what this instrument is very good at. Or if you're just interested in not killing your cell as quickly as you do on a confocal, try this one out. You'll also notice that these data that I'm presenting aren't noisy. We're collecting a lot of photons. And that's, I think, one of the more underappreciated things that Lightsheet delivers is far closer to the diffraction limit in resolution, because your signal to noise is so high. You're allowed to collect more photons before you damage the cell to the same extent. Your photon budget is effectively higher. And that means no more collecting data that are, grainy if you don't want to. You don't have to image every minute when you can image every second. It's a lot more data, but it's higher quality as well. We have totally open sourced the parts list for this. Alfred has done a great job of detailing the basic construction. I can share with you every part that you need to purchase from Thorlabs and others to build a microscope like this. And my buddy Joseph Berstowski at the NIH at the Twinbrook Imaging Facility took me up on this. And he, over the last year, has actually successfully built a replica of the one that I've done. But no hardware is good without a good software to run it. And I've been partnering with Nico Sturman at the UCSF Center for Advanced Light Microscopy. Nico, for those of you that don't know, is one of the main supporters of this open source microscope control software called Micromanager. And we built the hardware. We've also built a lot of the support software, mostly him. Building the support software to quickly acquire these data, render it in a way that a biologist is used to viewing, saving it. It's relatively seamless, but we're still making improvements. So if you do decide to build one of these yourself and use Micromanager, please make suggestions for features that we need to add, that sort of thing. Joe here studies, among other things, amoeba. These are Dictyostelium. For those of you that study Dicty, you know that it is very photosensitive. Joe's interested in the actin dynamics of this. What he's showing you is a 3D volume where we're sort of changing the view angle dynamically, but also showing you the evolution of like these actin comets. And he saw this one streak across the cell in a couple of seconds that he said he's never seen before, because we can't image these things in 3D this quickly. They will die on any other microscope besides the single objective light sheet. I think he told me to call this one the Mark of Zorro, which I like. Another buddy of mine, Torsten Whitman at the UCSF Biological Imaging Development Collaboration, has also built one. He, in this case, is showing you some cell division that's occurring. I'll try to represent these cells often as multiple orthogonal projections. So you're seeing in the lower right, like a traditional Z projection. I'll call it like the XY projection. Up on top here, it's the XZ projection. So you're seeing basically like this is away from the cover slip, and this is the YZ projection. So multiple orthogonal projections actually can be a pretty good, at least visualizer for what the cells are doing in 3D. At this point in the talk, I'm just going to show off some wonderful biology that I've been able to enable with a lot of the scientists at UCSF with this microscope. So what we have here are neutrophils that Patrick Zager has labeled with a plasma membrane. Patrick in Orion Weiner's group is very interested in how these basically like protrusions, these filopodial protrusions off of these cells, can evolve into more laminar lamellipodia-like protrusions. Like the T cells that I showed before, these cells are incredibly dynamic, but they're also very light sensitive. They do not make these really complex protrusions. If you are actively poisoning them, Patrick has to image these in 3D at 1 volume per second or more in order to see them. But he also needs to not kill them in the process. And he's actually one of the earliest adopters, not only of like my light sheet, but also the original one that Alfred built at Calico. So he's been a nice biological champion of like the wonders that this microscope can do. I had a scientist also from Dyke Mullins' group, Kyle DeMar. Kyle is very interested in how basically structural color formation occurs on things with scales like moths. So what you're looking at here is actually a section of a moth wing. What you might not be familiar with is that when the caterpillars are inside the chrysalis, they will grow their scales, and their scales grow in such a way that's determined by actin cables. And these actin cables, which you can see here fluorescently labeled, are parallel to the moth scales. They actually are they think the structural foundation that lay the groundwork for these really interesting complex like chitinous structures that give the moths their color. What you might not see from this movie is that this is a live moth. We took one image of it, but it is this wing is inside of the chrysalis. So Kyle, during his graduate work, figured out a way with Nipam Patel to image these developing cells inside of the moth wing. The moth lived after this imaging. They were able to have it hatch and grow, but this particular imaging basically requires a cover slip. Because the moth, and when it's inside of the chrysalis, is effectively encased in glass, chitin, and they oil couple the chitin to the cover slip, and so you end up with this effectively optically relatively clear path, but you only get one path into that. It's tricky to do with 2 lenses, but luckily we only need 1 lens at the sample, and with a clever trick that Alfred invented, which allows us to correct for the refractive index difference between the cells that are filled with cytoplasm and the glass that we're effectively imaging through. We're able to get deep diffraction limited images at subcellular resolution with this microscope. And this next one that I'll show you was taken by my buddy Jovan and Hernan Garcia's lab at UC Berkeley. Um, while I'll say not as visually striking as some of the ones that I showed you before, I'll highlight that it is by no means any less biologically relevant. What I'm showing you here is a relatively small section of a developing Drosophila embryo. And Hernan's lab is interested in statistical mechanic methods of basically describing how development occurs. They use the fruit fly embryo as a model for this, and what they're expressing here are particular mRNA transcripts in the developing embryo, and each of these bright spots is a single genomic locus. That at a particular point in the mitotic cycle, RNA polymerase will come in and basically transcribe these like mRNA transcripts, and these tiny little spots are effectively these mRNA transcripts like flying off. What I'll highlight for you is the timing of this. They imaged this at for an hour and a half, basically continuously, to watch these transcripts, rise and fall, not with bleaching, but this is just natively like what happens during the cell cycle of, there are different rates at which these mRNAs get transcribed during the cycle, and they're visualizing that in 3D live on a microscope, as easy as it is, to do on a confocal. So actually, I was teaching at the marine biological laboratory at the physiology course, and I was asleep. It was like 1 a.m. and Jovan texted me and wakes me up, and I got really but it was one of the best texts I've ever received, because Jovan basically said, dude, this is the best data that I've ever gotten first day. Like I have tried to solve this problem on many other microscopes. This was easy. I just brought it. We mounted it exactly the same as a confocal, and I got this data. So this is not even like the latest data that he's gotten. This is the first data, which I was incredibly proud of. Anyway, this is just on an emotional level, like why I do what I do. I really enjoy enabling biology by building better technology that lets us look at things that we've never been able to. And in that vein, I'll talk about a couple of interesting recent aspects of my work, and something that piqued my interest. This was published a couple of years ago, and this is a trick that when I saw it, I said, okay, I have to replicate this. I have to try this on a light sheet. So everything that I've been talking about is how do we, reduce the amount of bleaching and phototoxicity that we do to cells as we image? How do we increase the photon budget of the microscope? And what I'm looking for are strict improvements. I'm not looking for trade-offs. I'm not looking for like, oh, well, we increase the resolution by 100%, but we increase photobleaching by like 900%. No, I just want a strict improvement. That's why I'm trying to convince people to go from confocal to light sheet, is that it pretty much is a strict improvement. This is another, what I'll describe to you as another benefit that basically stacks on top of all of the others that light sheet gives you. So in this original publication from Ludvikova, they noticed that if you take green fluorescent protein, and not just GFP, but basically almost any green protein like YFP and neon green, wasabi clover. And normally, if you just excite GFP with, 470 nanometer light, like cyan light, it will bleach over time. But if you add in infrared light, in this case, 900 nanometers, and what I'm showing you, or what they're showing you here, is the 900 was only present in this red circle. It bleaches less, and not even slightly less, a lot less, like 2 to 10-fold less. This isn't like a barely noticeable effect. This is, I now have 2 to 10-fold more photons in GFP than I thought existed like last year. And this blew my mind. This seems simple enough to implement. A 900 nanometer light source isn't that hard. Cells are actually pretty tolerant of infrared. That's cells, most matter absorbs UV a lot more strongly than it absorbs infrared. You can even convince yourself of this by taking a white light source and putting your finger up to it, and you'll notice that the light that gets through your finger is red. Infrared gets through even like better. So if your cells aren't absorbing infrared, then okay, they're not probably going to be damaged that much by it. But if you've got a GFP in there and it seems to be protected by infrared, great, let's put that on a microscope. And not only does it protect GFP in a polyacrylamide gel, it also protects GFP in HeLa cells. It's not only like brighter for longer, they've also showed via like migration assays that these cells are healthier. By some definition of the word, they migrate more, we'll say regularly, than their counterparts that were not illuminated with infrared light. Cool. How do we get this on every microscope? What are some of the what are some of the potential like downsides to this? I'll go into this, but one thing I'll highlight is this is one of my favorite figures of the paper because there's very few times I think in biology where like you get a smoking gun, and this is one of them. So what they did is they plotted how well does GFP absorb infrared, in this case in the red, and they plotted it against if we illuminate like GFP with this wavelength, how much photo damage or how much less photo damage do we get? This is like they called the reduction in photobleaching, or RP, where this is basically how many fold more photons they got in this particular assay. And you notice they're the same curve. It's a pretty broad spectrum. GFP absorbs infrared, which you might not know, in the triplet state, which I'll go into in a minute, pretty strongly at 900, but even all the way out to like 800, it's a pretty strong absorber. And then you plot like, okay, well, how much does hitting GFP with this wavelength reduce photo damage? And it's it's the same curve. So this is the mechanism. So how does it actually do it? Well, this is their proposed mechanism. Normally, you have a ground state GFP. You will excite it with a cyan photon, and it will take one of the electrons in the bond that gets excited, and it will promote it to a higher energy state where it will stay for about 2 to 10 nanoseconds, depending on the fluorophore. So 2 to 10 billionths of a second before most of the time it will relax down to the ground state or some vibrational mode therein. And through heat loss, it will, basically emit a green photon, something that's like wavelength shifted more towards the red side of the spectrum. That's most of the time. That's the behavior we want. That is by no means the only behavior that we observe. Sometimes we get this thing called intersystem crossing, where it's often called the forbidden state transition, where the excited electron will flip its spin, and it will enter what we call the triplet state. At least the first triplet state. This is known in GFP to last for 5 milliseconds. So 5 million nanoseconds, like literally a million times longer than these singlet states will last. And it sits in this state. It is not bright, meaning that it doesn't, meaningfully contribute like green photons. It's not bleached, but it's often known to lead to the bleached state. We know that triplets absorb infrared because we can look at transient absorption, where we'll, flash in first some blue light and then look at how quickly does GFP, or how much does GFP absorb infrared and for how long. What we didn't know until this paper was what happens when GFP in the triplet state absorbs an infrared photon. We think that it is this infrared photon is promoting the GFP electron to a higher order triplet state, which is then energetically favorable for it to flip its spin back to the excited singlet state, where it basically immediately releases a green photon and goes back to the ground state. So what you're effectively doing is outcompeting the triplet state leading to the bleach state via interaction with things like dissolved molecular oxygen, where it will release a reactive oxygen species. We're outcompeting binding of the triplet with oxygen, or reaction of the triplet with oxygen, with absorbance of an infrared photon and driving it more towards this behavior that we'd like. So, all right, this came out 2 years ago. There is a delay in adoption of technological improvements that I'm trying to shorten. But again, why haven't we seen all of the microscopes just bolting on a 900 nanometer light source on it? And I'll highlight some of the caveats here. You need a lot of infrared light in order to get this to work. The data that they took suggests that in order to maximize the reduction in photobleaching effect, you need somewhere on the order of like 10 to the 3 or 10 to the 4 watts per square centimeter. This is about like 100 times more infrared light than you would normally use for excitation light. So you need a lot. And that's particularly tricky to achieve in a lot of cases. If I actually go back and I show you this, their original paper, you'll notice that they have a 43, roughly micron diameter spot. The amount of light that they put through this was about 1 watt in order to maximize the reduction in photobleaching. A 1 watt laser is, I'm not saying hard to come by, but that gets in the range of that is a little dangerous. I would be hesitant to turn on like a laser in my lab without all of the proper precautions, much less like bolt one onto the side of a microscope. And that's tricky when you're trying to scale up the field of view that you want. If I was trying to implement this on a wide field microscope and I wanted, we'll say 10-fold larger diameter, 43 microns is only a fraction of like 100x field of view that you often see. You need somewhere around 5 watts of light at your sample. But it works because, well, you're bathing all of your sample in light, not only in blue light, but also in infrared light. You're generating a bunch of triplets everywhere, not just in your focal plane, but also above and below your focal plane. And you're quenching them with the infrared because it also is effectively everywhere. But because you need such a bright light source, it becomes really hard to implement that on anything besides a fraction of 100x field of view because your light is defocused. It occupies a very large area, so you need a lot of intensity to get the correct, or you need a lot of power in order to get to the right intensity, which is power per unit area. On the other end of the, we'll say power spectrum, instantaneous excitation power is confocal, especially with laser scanning confocal. It's actually pretty trivial to get to the intensity of infrared that you need in order to get this effect to work. You only need a fraction of a milliwatt at the sample in order to basically saturate these triplets at absorbing like infrared. Here's the problem though. Despite the fact that you generate triplets of GFP at every focal plane, and you do this because you sweep your 488 across your sample constantly, the number of triplets that you generate at every focal plane in a linear modality is constant. So, I generate the same number of triplets way down here. I generate the same number of triplets at the focal plane and above the focal plane. However, the only place that I am effectively depleting the triplets before they're allowed to interact with molecular oxygen is at that focal plane, because that's the only place where my infrared is focused enough to have a high enough intensity to effectively outcompete reaction with oxygen. So I'm generating triplets everywhere, but I'm only depleting them in plane. So basically, this is not going to work with confocal, because if you have any diffusion of molecules between your in focus plane and your out of focus plane, your volumetric bleaching reduction is going to be negligible compared to what it would be without the infrared. And I believe this is why we haven't really seen like widespread adoption of this, is that I bet people have tried it. I bet it doesn't work. Light sheet, however, is this nice sweet spot between wide field and confocal. Instead of being point focused like it is in confocal, a light sheet is effectively a line focus. So you need more power than you need in a confocal because it's just a series of points, but you need a lot less power than you need in a wide field. I don't need a 1 watt laser for a fraction of a field of view. I can get away with a 200 milliwatt laser for the entire field of view, because at any one point in time, I'm only exciting a single like line or plane of my sample. What's nice about a light sheet is that I don't care about quenching out of plane triplets like I do in a confocal or a wide field, because I don't generate them in the first place, because it's a light sheet. I'm not exciting fluorophores outside of the field of view that I'm currently looking at. So basically, knowing this, we put a NIR laser in our light path. This costs less than $10,000, and most of that's the cost of the laser, and less than 1 day of my labor, where basically I just, alongside my visible path where all of my fiber coupled lasers go, I bolted down a laser, a beam expander, and a couple of mirrors with a dichroic here to marry the two beams together, which took way less than a day. And I made my microscope in the process over twice as good. What I have here are some agarose beads with EGFP embedded in them from my buddy Maria. And basically what I'm showing you here on the left is what happens when I image this thing volumetrically without infrared, and what happens when I image this thing volumetrically with infrared. Here's a plot of the bleaching rates. This is 2.5-fold better. I have now 2.5-fold more photons in GFP than I had last year. This is a fundamental break of a fundamental limit in light microscopy. We are photon starved. Every photon is precious. I've tried to show you how we'd be more efficient, or how we're more efficient with photons with a light sheet microscope. But I just happen to have found 2.5 more photons than I thought existed in the world's most popular like green protein. So for less than $10,000 and less than a day's worth of labor, you too can make your light sheet microscope over 2.5 times as good. But it will only work in light sheet. I'm pretty confident in saying, because of the reasons that I mentioned before. It is hard, if not dangerous, to do it in wide field, and I don't think it will work in confocal for the reasons that I talked about. But as cool as that is, I think there's a lot more money left on the table. And all evidence that I've seen points to this being able to be engineered. GFP, and not just GFP, but all of these other proteins like YFP Venus, Clover, exhibit this weird photophysics of I absorb infrared and I'm photoprotected by accident. And I'm pretty confident in saying that, because neither we, the scientists, nor nature selected for photoprotection by infrared. There effectively isn't infrared in nature down in the water column where the GFP is expressed by Aquaria Victoria. I can't say particularly like why it is, but it is. This is the classic hallmark of evolvability, because we've shown just, we'll say, wild type EGFP, like I haven't put any pressure on it, is not insensitive to infrared. And it seems to be also incredibly dependent on the primary sequence. There aren't that many mutations that are made to GFP to get it to a YFP, or wasabi to clover. Like the gap in the response between some of these is astonishing. And again, we didn't select for any of this. You rarely see like this large of a change in an effect just with a couple of point mutations. So given that the response of these proteins to infrared is incredibly dependent on the primary sequence, and we haven't put any pressure on it, I bet there is way more money left on the table. We have a GFP now that's between 2 and 5 times better. Like, how many more fold is left on the table? So this is basically an active area of interest for me and my buddy Andy. We're interested in finding fluorescent protein engineers that are excited about working on this problem with us, building instrumentation to actually go through the process of screening these mutants to find fluorophores that don't just give you twice as good performance, but another order of magnitude on top of that. So anyway, that's that's a lot of the recent work that I've been doing. Just to sort of summarize the talk, hopefully things that you took away from this. High numerical aperture single objective light sheet is the best modality that you, if you are a cell biologist studying live cells, live cells with a fluorescence microscope, you should be using this. It's gentle, it's easy, it's fast. You can further improve it by just bolting on an infrared light source, and you'll make your green channel over twice as good. It's easy, and I bet there's even more to be done. I bet there's even more like improvements that can be made by engineering, not the optical side, but the biological side. And that's that's what gets me up in the morning. So if you've enjoyed, if you've managed to stay awake, I appreciate it. Thanks for your attention. I can't thank enough all the people that have been really instrumental in helping me learn how to build this microscope. My buddy Andrew York has been just absolutely fantastic with that, as well as his colleagues Maria Garamo, Alfred Millet-Sicking, and Julia Lazari-Dean. My collaborator Nico Sturman, he is the software to my hardware. He makes the microscopes that I build work. People that have listened to me and built scopes sort of after my design, Joseph and Torsten, as well as all of the fantastic biologists that are willing to take a gamble of using a thing that I'm claiming is better, but I think actually have demonstrated that. All of the funding sources for the microscopes, several at UCSF, but also at the Biohub. And you can find my email and my social up there. This is not live. This is pre-recorded, but I should be live in the audience. And if you have any questions, please feel free to reach out to that or type it in the chat, and I'll try to answer it. Thank you all so much.
Thank you very much to Tanner for his presentation. It's now time for the live question and answer session. If you'd like to ask a question, please type it in now where it says type your questions here and then press send. We've got about 5 minutes for questions today, and to get started, we do have a question that came in a bit earlier. Tanner, it's a question from someone in the US, and they're saying, at a time when funding is so tight, what makes this technology worth the added investment in addition to a conventional fluorescence microscope? What are your thoughts on that?
I appreciate that. It's something that I think about a lot. I try to be as useful to as many people as possible. I haven't happened across many improvements that are this large, where you get effectively 2.5-fold more photons with no trade-off for this low effort. Meaning less than $10,000 US, less than a day's worth of labor. It's certainly an investment, but I don't know of any other improvements that are that large of an improvement, 200 or 150% increase for that low of an effort. If there are, I'd love to know them. I think that if you've already got a light sheet microscope, this is a fantastic and low cost addition on top of it. If you're a core that is thinking about, okay, what's the next microscope to build, for a lot less than a pretty high-end confocal microscope, you could build a single objective light sheet. That's certainly a lot more upfront cost than just adding infrared to an existing light sheet. But I'll argue that it's a larger delta as well. You get hundreds-fold less damage and 100-fold faster for basically the same price. Now that comes at the cost of building expertise and that sort of thing. But I always love teaching people, so I'm going to try to make that cost as low as possible.
Great, thank you. Someone else is asking, does near-infrared co-illumination work in red fluorescent proteins?
This is super wild, sort of. In the original paper from Ludvikova, they showed that with if you express M. cherry and E. coli, and you plate those E. coli on LB plus agar, and you co-illuminate with infrared, they bleach slower, just like GFP. Great. It's not that straightforward. If you plate the E. coli on PBS plus agar, and you co-illuminate with infrared, they bleach faster. So it's actually, you're doing more damage to M. cherry than you were without it. And I've actually independently replicated this with M. scarlet and M. cherry, that I can get it to bleach faster in PBS than without infrared. As suboptimal as that is, I'm not too worried about it. I think just like green proteins, there's a lot of money left on the table. Clearly, this is very like primary sequence dependent. I bet we can do a mutation screen to figure out what mutants do we need to stack up in order to, flip this effect. Because the hardest thing is engineering a response at all. The easier thing is figuring out how to optimize the response. And clearly, they demonstrate a response. So, in a sense, it's bad, but there's even more money left on the table for red proteins than there are for green.
Great, thank you. We have another question that's come in. If NIR or near infrared co-illumination is good for green proteins, why does multiphoton each?
Sorry, I think either I or you cut out there. This is about the two photon question.
We froze a bit.
Yes. So two photon is interesting. It's very rarely discussed in the literature, but it's very widely known among people that use two photon that it just bleaches more quickly than in a single photon modality like confocal or widefield or light sheet. And there isn't a good consensus why, which I think is why it's not talked a lot about in the literature. It's just something that people know. But if you come into this field thinking that your GFP has the same number of photons on a two photon as it does on a confocal, you're just wrong, and you're off by about an order of magnitude. Why that is, I think a lot of this recent research has pointed to. If there's a lot of focused NIR at the focal point of a two photon microscope. The whole point is that you absorb multiple photons within about a femtosecond of each other. If a green protein gets promoted to the triplet state, which is, let's say, a 1 in 100 fold or 1 in 100 chance that it does when it gets excited, and it absorbs an infrared photon, it'll go to the excited triplet state. But if it then absorbs another infrared photon on top of that, which is extremely likely in a two photon case, and I'll say at least a lot less likely in the light sheet NIR illumination case. In the two photon case, it keeps absorbing infrared photons and probably gets promoted to a really high energetic state that I suspect is much less stable and much more likely to go to the bleach state. So it might even be that in two photon, if you go into the triplet state, it's a near certainty that you get bleached, rather than in a one photon case where it's possible, but maybe a lot less likely. Again, these are suspicions rather than like I have tested this. Transient state absorption of fluorescent proteins is hard. I can only point to like what we sort of know just by looking at green proteins under a two photon microscope.
Great, thank you. That's all we have time for in terms of questions today. But if anyone has sent in a question that hasn't been answered live, we'll make sure that we send you a response to the contact details that you've provided. So everything will be answered. So I'd just like to thank Tanner again today for his presentation and for answering our questions today. And I'd like to thank our sponsor, Hamamatsu, and of course you, the audience, for tuning into this webcast today. And remember, you can access upcoming and on-demand Nature Portfolio webcasts at www.nature.com/natportwebcasts or via the QR code, which hopefully is on the screen now. Thank you for watching, and I hope you can join us again soon.